Um, so today I want to introduce you all to Dr. Aleeza Gerstein, who's visiting with us from the University of Manitoba as a new associate professor, having just gotten tenure this year, transitioning from assistant. So congratulations.
Yep.
She started her research career really focused on questions as part of a trainee in Sarah Otto's group at UBC, where a lot of this focused around the idea of ploidy and how that affects evolutionary paths and adaptation, but also investigated a lot of really interesting concepts like Haldane sieve and how you can bypass Haldane sieve through loss of heterozygosity in different regions of the genome. From Sarah Otto's lab, she ended up postdocing in Judy Berman's group at the University of Minnesota, where she continued this same kind of concept of karyotypic variation and how it impacts phenotypes. And I spent some time in Kirsten Nielsen's group, also at the University of Minnesota. From there, she went on and initiated her own faculty position in 2018. A lot of her lab's work is centered on the same questions around adaptation and evolutionary processes, oftentimes focused on microbial responses to drugs, mostly around fungal drug exposures. As part of this work, she's done incredibly well, published a number of papers and finding a lot of support for her work, and then was also awarded a very prestigious Azrieli Global Fellows position within the fungal kingdoms group. And I think you just finished your term on that.
Yep.
Great. So with that, I'll turn it over to Aleeza, who's going to be telling us about a little bit of a different topic, I think, than the antifungal drugs, but
we'll get there eventually. Okay. We'll see if we get there eventually. Okay, so first of all, I had a wonderful day here. I have never been here before, which is kind of weird. I was a postdoc in Minneapolis, and I've known about sort of this department, or not department, I guess this genetics group at Wisconsin my whole career. And so it's really fun to spend the day here, sort of intermixing between people that I've met throughout my path in academia, starting in like ecology, evolution, theory, and then now moving into clinical microbiology. My faculty position is bizarre in that I'm cross-appointed in a microbiology department and a statistics department. I am not what I would never call myself a theoretical statistician, but as an evolutionary biologist or ecologist, turns out we know a lot more statistics than other people in microbiology. And so I'm going to pull from what the stats folks do. And instead of standing here and talking for 50 minutes, and then having you ask me questions at the end, I think in this 2024, when if your brains are all like my brain, there's no way I can pay attention for 50 minutes to even the most enthralling speaker. And so the stats folks interrupt. They give seminars and people will put up their hands and be like, whoa, 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 what did you just say back there? Can you explain yourself better? And to show you how serious I am about this, I pilfered my children's Halloween candy, and I've brought you Canadian chocolate bars. So if you are so brave to ask me a question in the middle and interrupt me at the end, you can come up and take your choice of Canadian chocolate. Coffee Crisp and Smarties, which are different than your Smarties. Our Smarties have candy and chocolate combined. Cool? Yeah? Okay. Yeah, great. So thanks again, Matt, for the invitation, for putting my name forward, I guess. Yeah, today I'm going to tell you about some parts of my research that my lab has been doing on human fungal pathogens that we study through the lens of evolutionary genetics. This is my group, so these are my graduate students and my technician, and then very important person, Dr. Vanessa Colloquium, who's an OBGYN and clinical researcher at the University of Manitoba, and Rebecca Kukaruds, who's now a genetic counselor, but was a former lab tech who facilitated a lot of what I'm going to talk about today as well. Okay, so I've started almost every talk I've given for a really long time with this picture. This is from the Field Museum in Chicago, and this is, Darwin's quote, "From so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved." And I think this sort of captures what sparked my interest in biology, especially evolutionary biology in the first place, is just thinking about how do organisms end up looking the way they end up, and how are things sort of matched to the environments that they find themselves in? So what happens in evolutionary time if we go out into the world and look at a forest or a grassland or an ocean, and if we care about microbes or macrofauna, why do things look the way they are, and why are they where they are? And I got turned onto fungal microbes sort of by happenstance and chance, but I think They are the coolest for lots of reasons. And one of those reasons is because if we look at the phylogeny of fungi, we see plant pathogens, human pathogens. This is calling them binary. They're not always binary, but whatever. You can see they're sort of spread out throughout. There are some groups like the Canada group, which I'm going to talk about today, which are the sort of clusters of human fungal pathogens, although this is not at all the complete phylogeny, and there's lots of other species that are not human fungal pathogens in there. We see this sort of wide sort of spreading of human and fungal plant pathogens. It's not like there's like one trait that happened one time that like branched off to human fungal pathogens, and another one that branched off to plant fungal pathogens. For microbes, they sort of like look diverse, if you will. The two species we work on most in my lab are Candida albicans and what's called here Candida glabrata, but it should really called Nicosaomyces glabratus. There's a huge debate in the fungal field right now about whether or not we should be renaming species because clinicians deal with candidiasis. And so they want everything to be called Candida. But okay, so how we can look at them easily is on something called chrome agar, where Candida albicans is green and Nicosaomyces glabrata shows up as pink. This is sort of the distance of relatedness. So there's Cerevisiae always at the top, glabrata closer to Cerevisiae, and then albicans down here. And this substitution doesn't really mean anything unless you speak those specific numbers. But here's the phylogeny. So there's 200 million years of evolution that separate the Nicosaomyces Saccharomyces cluster from the albicans up at the top. And if you compare this relatedness to other things that we might think about, you know, if you look between humans and glabrata, that's humans to zebrafish, and then down to like sponges and comb jellyfish. And so although we call all these things yeasts, and we, I think in our heads, lump them together as well, they're all just yeast. They all look morphologically really similar. There's actually tons of evolution that's happened. And so although I'm not really going to talk about the difference between glabrata and albicans today, I think it's a cool sort of comparative system where we grow them in the exact same media, in the exact same test tubes, in the exact same incubators. They look superficially very, very similar, but we can ask really cool questions about their genomes and how evolution works in these different species. And so a lot of what I've been interested in studying is this sort of diversity writ small and writ large. And so Candida albicans has lots of diversity within the species. So they grow in diverse morphologies, so they can grow as yeast. They can also grow as pseudohyphae or as hyphae. They can grow as planktonic cells. They can grow in biofilms. They grow as what's called white, or like typical cells, I guess I would call them, or they can grow as opaque cells, which are the mating cells, or the cells that are capable of mating. You can look at them on plates where the opaque cells are not opaque, but pink. They can grow in different ploidy states. So naturally, albicans is mostly diploid heterozygous. There's reports in the literature about haploids. You can kind of do some trickery to get them to be haploids. They don't like being haploid. They don't stay haploid for very long. But we definitely see tetraploids. We see aneuploids, just like we see in Cerevisiae. They can exist in that phase for quite a while. They can mate, and they can do what's called parisexual reproduction, where they kind of lose chromosomes mitotically, which if you're interested in aneuploidy, which I am, it's kind of a cool system. There's sort of lots of variation going on within the species. And the reason that got me into albicans in the first place, so coming from Cerevisiae in graduate school, you all deal with wild Cerevisiae strains, but not many people deal with wild Cerevisiae strains. And I really like the idea of studying a microbe where we knew where it was coming from, and we knew the niche where the isolate came from. And albicans grows like everywhere on the human body. So as a commensal, it grows in the oral cavity, in the nasal cavity, on the skin, and the GI tract, and the vaginal tract, where it just exists as part of the microbiome, either as a commensal, maybe it's doing something beneficial. Lots of questions to be figured out. These niches vary wildly in terms of temperature, pH, nutrients, the other microbes that they encounter. And then it can also grow as pathogen in the exact same places where it's growing as quote unquote a commensal. And what does it mean to grow as a pathogen? Well, it means it causes inflammation or it causes disease, however you want to define that. And so again, same places. Some of this is caused by, weird things happening. So like they'll form biofilms on devices. If you're in the hospital, you get like a central line put in, they'll form a biofilm and that causes disease. We see some cases of immune suppression. You can have an uptick in sort of fungal infections. But in some cases, we have kind of no idea what causes them to switch from this sort of commensal life cycle or existing in your body without causing inflammation and disease to being a problem, to saying, hey, I'm here, you need to pay attention to me now. And so that, I think, as an evolutionary biologist also is really cool. How are they doing this? Why are they doing this? And is it something special about Candida albicans specifically that makes them different than other microbes? So I'm going to tell you about one study that's looked at variation in the oral microbiome. So this is work out of Teresa O'Meara's lab. It was published in PLOS Biology a couple of years ago. They got a bunch of undergrads, they sampled them, they got isolates from their oral microbiome and some of them rectal microbiome as well. So these on the top, this is just showing differences in growth from these different isolates. So this is 30 degrees, everything grows as yeast, that's pretty well known. 37 degrees in serum is known to influence or to cause most isolates to grow filamentously, but you can see one here that isn't really growing filamentously. And then 42 degrees also typically filamentous growth, but again, one that's really not growing filamentously. They looked at invasion in these different environments. You can see that environment sort of shifts the distribution, but the thing to take away is there is a distribution, again, of these oral isolates in these different environments. So we're seeing variation between people in what their oral commensal, these are not disease, commensal oral isolates are doing. They see some cases of variation within the oral population. So this is a UMAP plot, which some people have lots of feels about, but basically it's a way to take lots of different phenotypes and map them onto 2D space. And so all you need to know is when you see isolates or you see numbers in different places, those are isolates coming from the same people that phenotypically look very different. So this is two isolates growing contemporaneously at the same time in the same population that look quite different. So there's standing genetic variation or standing phenotypic variation in these healthy oral Candida populations, if you will. And one of the big takeaways from their paper was that this sort of commensalism, so oral isolates just growing without you knowing that they're there, does not seem to select against infection. So if they use the Scalaria infection model, most of the isolates are sort of above that competitive index. And so they're all still able to cause an infection. If you ask them to, again, with some variation, the color is just oral versus rectal. Okay, so what does it mean to be a pathogen? Does it mean anything? Is there anything different about pathogens? How are they doing it? Why are they doing it? And so the question that I've now realized we're actually studying in my lab after 7 years or 6 years is the question, sounds really simple, is there adaptation? So in all these different niches where you see all these different populations that have wildly different abiotic and biotic pressures, are we seeing adaptation happen? And so I'm going to tell you about some of the work we've done, and I can tell you that the end result is going to be, we're still working on it. But I think thinking through even what the nomenclature should be when we're talking about these microbial populations is not set in stone. So people talk about adaptation a lot without necessarily meaning allele frequency changes over time that are beneficial. And showing that there are allele frequency changes over time that are beneficial is not necessarily trivial. Again, feel free to ask questions at any. Okay, so this is sort of the framework. We're going to use ecology and evolution to try to understand human infection, but I also think we can use human infection as an eco-evo model because we're pulling these populations out of different niches in a way that I don't think we can necessarily do in non-human associated populations where we can where we take swabs or we take a sample and we know exactly who it came from, where it came from, when it came from. Okay, so the disease or the context in which we're doing a lot of this work is vulvovaginal candidiasis, otherwise known as a yeast infection. So this is the one that gets the nomenclature of yeast infection. It's super common. What it is not is super commonly studied. So 75% of people with a vagina will get a yeast infection at some point in their lives. They cause significant financial and emotional strain on the people who get them. It causes physical and mental discomfort. It causes a loss of productivity. Most people don't talk about it, right? It's like a women's health issue and it's icky. So we talk about it because it's really common. So that's 75%. Of that 75%, 8 or 9% will have recurrent symptomatic cycles. So that's 3 or 4 or more times a year where you're experiencing these symptoms. And it is essentially a chronic condition for which there is essentially no cure. Sometimes it's alleviated at menopause. Sometimes it's alleviated sort of randomly within your lifespan. But essentially, we don't know necessarily. So what do we know? You can have people with vaginas who are uncolonized by Candida albicans, at least by culture. So if you take a vaginal swab and you put it on your plate, 20% of people who don't have disease will be, I would say, commensal or asymptomatic. If you do ASV, so if you do like for ITS sequencing, it looks like it's a much higher proportion. So they're just below the culture detection limit. And if you do temporal sampling, it also, yeah. So there's some temporal variation in whether or not healthy people have Candida albicans there. You have this huge group of people who have uncomplicated yeast infections. So that's like, you have symptoms, you go to the doctor or you go to the pharmacy, you take a drug, typically fluconazole, over the counter, symptoms go away, you go on with your life, and hopefully that's the end of it. But 8 or 9% get recurrent symptoms. Between the symptom cycles, whether or not the yeast completely go away or turn asymptomatic is kind of unknown. Yeah. And I'm being really careful to call this recurrent symptoms, not recurrent infection. Because again, we don't actually know what's happening between these cycles of symptoms. Okay, so what I'm going to tell you about today is 2 stories, or 2, I guess, programs. I'm going to call them programs from my lab that I stole from a friend who called their work in a totally different context from bedside to lab bench. And this is mostly I'm going to focus on these mismatches between evolutionary predictions and observations because I think that's interesting space to work in. And like I said, this is done in collaboration with an OBGYN, and a lot of this is motivated by conversations with her about what's just unknown in her world and things that she sees. And so the first one I'm going to tell you about is a sort of idiopathic recurrence where we're doing clinical ecological sampling. And then the second one, I'm going to tell you about some experimental evolution work. All right. So this is our infection triad. We have humans at the top, our bacteria, and our yeast. And so we're looking at these recurrent cycles. We know something about some human genetic on the human side of things that does influence whether people are prone to getting recurrent symptoms or not. So we know it's this non-protective inflammatory response, so it's neutrophils, the innate immune system. There's a small number of genetic variants in humans that have been identified in what I would call very low-powered studies, but there are some that are known. And then we know about certain treatments that cause spikes in symptoms. So things like if you take antibiotics, which affects your bacterial microbiome, sodium glucose 2 transporters, so it's for diabetes, more prone to getting them, people who are pregnant, which is a slightly dampened immune system, more prone to getting them. But as far as the literature can tell, at least half of cases are idiopathic. So half the time we have no idea why someone is predisposed to getting these recurrent cycles of symptoms or not. On the bacterial side, lactobacilli proportion, maybe. And so we think that a healthy vaginal microbiome is dominated by a lactobacillus. There's multiple species of lactobacillus. There's also more diverse microbiomes, and it's definitely not a clear one-to-one. You have this species dominant or you don't. You have symptoms or you don't. We're working on it. We'll get there eventually to look at the correlations, but I'm not going to talk about it today. And then on the fungal side, Candida albicans is a major player. Candida africanus is the closely related species to Candida albicans. It's actually still included in the albicans phylogenetic trees. It's called clade 13. It should be its own species. And then Nacosomyces glabratus, this completely, I would say unrelated, really, yeast species, also causes infections and is increasing in some places in frequency. The clinicians treat this all as candidiasis. There is nothing, there is some innate drug resistance in glabratus, so they might change the drugs that they're prescribing, but from their perspective, it's all candidiasis, it's all vulvovaginal candidiasis. Okay, so the first thing we set up to answer was to look at ecological sort of sourcing dynamics within the body, to say how related are the populations at different places. And so this is my like microbiologist view of what the human body looks like, or my ecologist view. So we have different patches. I've drawn the lines where I think they should be drawn in terms of how migration might happen. And so that's what we're looking at. And so to look at this, to start looking at this, again, we're asking, is it evolution in action in the human body? So can we get a signature of adaptation to the vaginal environment? Is there something specific about those isolates or those populations? I don't think I need to for this audience, but evolution does not mean adaptation. We want to see beneficial. And so there's sort of two things that I've been thinking about. So one is that selection requires genetic diversity, right? So you need diversity to act on, or evolution to work. And then adaptation also requires selection to overcome drift and migration. And so this is just sort of very simplistic. That is sort of affected by the effective population size or the population size, and then the migration rate scaled by beneficial mutations. So we set up with Vanessa this clinical sampling program. She has a once-a-month yeast clinic where she's referred people who've seen their primary care physician enough with these recurrent cycles, and their primary care physician is like, I don't know how to help you any further. Here's a referral to Vanessa. They come into our clinic and we enroll people in this sort of 1-month sampling program. In the clinic, we get vaginal, rectal, oral, and skin swabs. We send them home with kits to do 1 month of self-sampling, so 3 times a week, vaginal self-sampling, and then they again swab vaginal, oral, rectal, and skin sites. We take the swab-elutes, we put them in PBS, and then we plate. And when we get isolates, we take up to 24 isolates, which is really different than what they would do in a clinical microbiology lab, where they would say, you have Candida albicans, and they would pick a colony. Unless the colonies look wildly different, they would only take 1. So we're interested in variation. We have almost rapidly filled up our freezer, which is a problem, but I think it's, yeah, worth it. To this point, we have 28 participants that have these time point samples. We have 10 participants from a pilot study that just did those day 1 samples in just vaginal and rectal. And then we're also starting to get asymptomatics. This is the point of the talk where I tell you, do not try to start a clinical program during a global pandemic. It's slow, so it goes. Okay, this is what we thought we were gonna get before we started. So we were like, okay, they're gonna come into the clinic. They're gonna have an infection or symptoms. They're gonna get prescribed drugs. We're going to see the yeast population high. They're going to start taking drugs. Population is going to crash. And then we can look to see what happens, in the swab-elute, either in the bacteria or, see if the albicans population disappears below detection, whatever. This is what we thought. And when we looked, this is what we actually have. So these are all of our participants that have at all cultured yeast on a plate. There's one more that we just got, but it's not in here. All the rest have no yeast at all on plates, which makes sense because they were referred specialists. They don't get to choose when they see that specialist. They got this appointment who knows how long ago. And so we have a lot of zeros. But even of the ones that came into the clinic, we have a bunch that came into the clinic with yeast populations that were high and stayed high the whole way along. We have a couple that came in high, got prescribed drugs, population crashed. And then we have some that came in low and then went up in that month that we just kind of by chance hit them during that month. So things that look like this. One of the things that I'm not going to say, I'm just going to say it and then that's all I'm going to say about it, is that the symptoms don't match the yeast population. So we also send them home with calendars where they track symptoms for us. And we see some of these folks at the top that are having like raging high yeast populations do not report symptoms. And some of the zeros that I'm not showing you do report symptoms. Yeah. Mhm. No. So we're not it's not a like intervention. It's not a clinical trial. And so we're not doing any like they are getting treatment if they were going to get treatment. They're not getting treatment if they were going to get treatment. They're getting prescribed whatever they were going to get prescribed without us. Yeah. Yeah. So there are differences. And so we set out to ask what a sort of simple question, which is like, what is the nucleotide diversity? What is the population level diversity? Oh, yeah. No. No, because even if you look at just the day zeros, so people just coming in, it doesn't match. Yeah. And I should say like the self swabs versus the clinician swabs, there's lots of literature that says there's no bias in who's actually taking the swabs in terms of what you get culture of population. Yeah. Yeah, so they kept well in the freezer, -20, and we did in our pilot studies, we did pilot work to be like, do we see a difference at least as best we can in the number of colonies or the drug resistance phenotypes of in -20 versus not? And the answer was no. They're stored in glycerol at -20, and then the clinical ones we get like the same day and we process them, and then they go straight into glycerol at -70. Yeah, that's a good question. Okay, so asking the question of what is the variation in these populations? We picked so far all the red dots. So we picked the time points that were most extreme, and then 5 populations from the D0 samples, and then picked 6 isolates from each of those populations. We calculate nucleotide diversity with this program called PIXI. So what we're looking at is basically pairwise differences between genotypes. So if you have 4 different isolates, you're looking at 1 versus 2, 1 versus 3, 1 versus 4, et cetera, et cetera. The like easy way of doing this just takes sort of the total number of comparisons divided by the number of changes. So this is TG, this is CG, and it just divides it all by 6. And the nice thing about PIXI is it accounts for like the short read sequencing where we see sometimes these like missing data. And so it counts for missing data in the calculation. So it's just a more accurate way of measuring it. That's what we do. This is the distribution of pi that we see across these samples. This is again 12 samples. Okay, so we see variation. It kind of seems to map the trajectories of the patterns we see. So the low variation sample is one that started off low. We saw it rise and then it sort of rapidly went down. So potentially indicative of this sort of like rapid population expansion. Same thing here. And then these sort of higher ones are either, we don't know what happened before they came into the clinic, but presumably they've been high for a while, or these sort of populations that were high and stayed high. And so we're seeing that this matches to these trajectories we see over time. So, okay, that's nice to know that our swabs are able to pick up what looks like biology in terms of the population. If we look at the co-occurrence of yeast at multiple sites, so this is just all the vaginal positive ones, we see that a bunch of them are rectal positive, and then much smaller number are oral or skin positive. Again, this is by culture, but we do have some samples, sort of every combination you could imagine of multiple pits at multiple places. So if we just sort of look what that looks like, the rectal populations have a much higher proportion of people that are rectal positive than healthy undergrads, that oral study. The oral and the skin seem to be more similar to sort of healthy people. So note. And so the question we're asking now is like, is there migration between the sites? And so we took the rectal isolates and the vaginal isolates from 4 people, 3 from albicans, 1 from glabratus. Again, 12 isolates, and asked the question, like, what do those populations look like? How similar are they? So here I'm just plotting pairwise SNP differences. So again, this is 1 against 2, 1 against 3, et cetera. Here's the distribution. So this is rectal against rectal. This is vaginal against vaginal. we can see that there's the same level of diversity in both the vaginal population and the rectal population. And then if we play the game where we take one rectal isolate and one vaginal isolate, if they're from different populations, that should go way up, which is not at all what we see. And so based on this measure, these look like the same population. If you compare two vaginal isolates, they're just as similar or just as different as if you take a vaginal and the rectal isolate. And these are like internal rectal swabs. So it looks like there's considerable migration, at least during a symptomatic relapse. If we map these on a phylogeny, these are all closely related. So like there's very little bootstrap support for most of these, but if it's just by chance and not biased, if you look at where the triangles are and where the squares are, they're like interwoven, there's overlap between the two populations. And so we did this, and then we're like, okay, but what does it mean? Like, is this higher or lower than what we expect? What do we actually expect? And so we dug into the literature. Oh, sorry, one more thing. This is just plotting pi, I guess essentially the same thing as the pairwise SNP differences. The glabratus populations are way lower diversity than the albicans, but otherwise very similar. And so, yeah, is this higher or lower than we expect? Very few studies in the literature have actually looked at different isolates from the same population at the same time point. We found two for albicans. So one was that PLOS Bio oral paper, and then we found another one that also looked at oral isolates from 3 different people. And so we see, by and large, sort of very similar levels of diversity in our theoretically or population up-down sort of crashing populations that you see in healthy oral communities. There's a couple of people who had wildly different isolates in their oral communities, sort of low population size, but I think this speaks to like how little data there actually is on the ecology of microbial populations, even in contexts that we care about. And then when we look at the glabratus ones, the work was only done in bloodstream infections, and so ours seem higher than what you expect in a bloodstream, which we sort of think no is like really a small number of cells get into the bloodstream and then have this rapid population expansion. And so based on this, it kind of tells us that's not the regime that we're in these infections, because there's like one population, one cell feeding these isolates. So we think that's really interesting. I believe Matt Lab is working on sort of similar questions, and we would love to have more data in different infection contexts or different just microbial body population contexts to be able to sort of situate where we're at and how much diversity we have. So the interesting thing about albicans is that, so this is the phylogeny. We've mapped just the vaginal in purple and the rectal or stool samples in orange. This up here at the top, that's Candida africana. So they're almost all vaginal isolates. We have a small number that are not, but by and large, it seems to be like vaginal specialists. We've whole genome sequenced 73 other isolates from Winnipeg. So those are all these dots all around here. So we have Winnipeg isolates sort of throughout this global phylogeny. And then these arrows are our Thrive isolates. That's what we call our research program. And so they're also more or less throughout, but they are clustered in what's currently called clade 1, which we think is interesting. And we think it's really interesting because there's a sort of lore in the albicans field that albicans lacks geographic substructure. And so if you go to like Candida auris, we see entire clades of the tree are like from Africa or from North America or from Europe, and that's not what we see here at all. They're really sort of a scatter shot. But if you look, we're seeing these Winnipeg isolates sitting right beside each other in the phylogeny. And so here we have like a knee isolate from 2012 right beside a vaginal isolate from 2022. Okay, that looks like geographic substructuring to me. And then we also see, and here also, so blood isolates beside our vaginal isolate. And then there's also this lore that it lacks isolate type substructure. And so if you plot sort of where isolates are coming from, again, it just looks like a scatter of colors. But we have cases like this, where we have these 3 Winnipeg vaginal isolates and beside vaginal isolates from Brazil, China, and Morocco. And so it does look like at this like granular level, there is sort of potential for maybe we can pick up on what the genotypes are that are under selection. That's something my PhD student is working on. Yeah. Yeah, so so you pick 12 isolates from the vaginal and the rectal, like swabs that were taken on the same day from the same person. We also have data I'm not showing because it's still in the works, but we picked isolates from other places in the body that are at the same time, and they all look to be very sort of similar in terms of relatedness. And so it looks like you have sort of one dominant genotype with some variation. And the amount of variation is, and it's not nothing, right? And so it's the question of whether or not we can pick up whether the variation that's there is just drift or whether it's like actual sort of niche specialization. And that's what we're working on essentially. Yeah, so I started by saying, like, is there scope for yeast adaptation to the vaginal environment? So there is diversity. Cool. Has selection overcome drift and migration? I think it's still an open sort of question. We certainly, I mean, we know that the populations cycle, and we know that now there's like lots of migration, but is selection in here enough to overcome these things is what we're working on now. We're going to do this in this sort of phylogenetic context by pulling in all these other isolates, both local and global, to sort of look at the genomic signature to see if we can pick something up. Cool. Is there any questions about any of this? Yeah. I mean, they're very, like the proximity is obviously very high. So I think there's, I hate saying like hygiene because I think that, puts onus on people, but there is probably some hygiene practices that are moving things around. Yeah, and so what we see is this pilot. So we think that there is potential to say like, yes, selection is happening. There's these phylogenetic clustering, but I didn't show you the phenotypic data because it essentially shows no variation. So we see genetic, there's 500 to 5,000 SNPs that vary among these strains. But we see essentially no variation in drug resistance or invasion ability. And so as an evolution biologist, you're like, well, these people are taking drugs repeatedly and frequently. So obviously the populations are going to become drug resistant, but they don't. So we see some cases of drug resistance in vulvovaginal candidiasis, but like very, very little. And so they're not adapting to the drugs. And so we can ask the question of like, what is the selective pressure? And we have, as part of Thrive, a really great collaborator who's also at the University of Manitoba who works on bacterial vaginosis. And so she has like a vaginal epithelial model. And so we're gonna hopefully soon start co-culturing things with vaginal epithelial cells to try and get a measure of fitness with that. Okay, so now I'm going to tell you about drug resistance. So we're going to talk about some experimental evolution, which is something I've been doing a long time. And again, this is sort of through this lens of what actually needs to be done in this space. And so this is the Society of Gynecologists Canada Clinical Practice Guidelines for uncomplicated, recurrent, and non-albicans vulvovaginal candidiasis. And again, this is because these non-albican species have this intrinsic resistance to fluconazole, which is our sort of favorite drug to treat fungal infections in general, or albicans infections in general. Fluconazole exists as an over-the-counter medication, along with some other things. And so we see it being used in uncomplicated and recurrent vulvovaginal candidiasis. But then the other one that shows up here multiple times is boric acid. And Vanessa, in our like very first coffee conversation, was like, you should study boric acid. It works really well. When I see women where they've recurred on fluconazole, I prescribe boric acid, but we don't know the mode of action, and they have to go to a compounding pharmacy to get it, or to Amazon. And so boric acid has been studied for a really long time. From the 1800s. This is a paper from 1981, which talks about it as a dangerous drug of little value. This is also from 1981. 14-day intravaginal gelatin capsules. Cure rates were 92% at 7 to 10 days and 72% at 30 days. Drug quickly alleviated the signs and symptoms. There was no untoward side effects. Several gynecological features were not affected. clinician find success with it. Not to trust everything on the internet, but if you go on Amazon and look up boric acid, 75,000 people have commented on this 10,000, 11,000, 3,500 on these boric acid vaginal suppositories. All those people cannot be wrong. And if you read the comments, it is like heartbreaking because it is people saying, I have suffered for 10 years and a friend just told me about this. And why didn't I know about this 10 years ago? Why? Right? And it's not that expensive. So we want to study it. And we thought we would be like, you know, it'll be so easy. We'll just run the omics playbook. We'll look for isolates that are resistant. We'll evolve resistance in the lab. We'll figure out what it's targeting. Yeah. So we're like, we'll just evolve all the things. That's our favorite thing to do. Let's do it. Easy. It'll be easy, they said. So we took 8 C. albicans strains. We did this work before we had our own sort of vaginal isolates. So these are some of the standard yeast isolates that people work with in Candida albicans. We took 8 different strains, 12 replicates, and so we have a total of 96 replicates evolved to increasing levels of boric acid. So we start them in a low level, and then we're passaging them at an OD of 0.3. So that's like when they're hitting exponential phase, and we passage them to the next level of drug. So this is what the survival curve looked like. So this is, we took 12 replicates, and right away we were like, oh, they're going extinct in this drug. This is our second attempt at it. And our first attempt, everything went extinct after 4 passages. Then we tried to like really like baby them, like we're giving them like lots of passages before we increase the drug concentration. But yeah, they all sort of end up extinct eventually. And this is again a survival curve. So this is like when you see a dip, that's a replicate that went extinct, or multiple replicates that went extinct. Almost after we transfer them, immediately after we transfer them into a new level of drug. So this is increasing drug across the top there. This is for one of those 8 strain backgrounds. Here's all of the 8 strain backgrounds. So they're separated into 2 different plots based on what happened after we transfer them from 1.5. So these ones we had to like, we just went up by like 0.25 mg. Yeah. Great question, and I can't answer that. I can tell you that like the initial MICs of these isolates is real tight. So they are all like 0.8 essentially mgs per mil. Like there's almost no variation. I should have had it at one point, but if you look at like 100 isolates and how they vary in fluconazole, you'll see this like nice sort of distribution. In boric acid, it was like they were all right there. And so we just took basically one step below the MIC because the goal is to evolve resistance. Yeah. And so each of these dots, I should say, is like a transfer in that level of drug. And so these ones all, if we put them straight from 1.5 into 2, everything died. These ones, we could take them from 1.5 and put them into 2 and things still survived. And so that's why it's split. But essentially, number of transfers on the bottom and everything is extinct by like 3. Just really not ramping it up that much from where they started from when you're used to working in drugs that you can like ramp up logarithmically. This is like small linear changes. But okay, whatever. So we pulled them out of things that they were growing in better. And so we decided to focus on 2 of the replicates from here and 2 of the strains from here. And we just pulled out these sort of last surviving replicates. And then let's do the thing, see what they're doing, see what they can do. And what we found was that resistance nor tolerance, which is a sort of ability to grow slowly in drug, if you give them enough time, increased at all. And so here's our 4 strain backgrounds, our multiple evolved replicates, and like they're not more resistant than the ancestors were, which is like they're not playing along. Why? Yeah. So they didn't get better. We see small improvements in growth ability. This is no drug. This is like RPMI, which is our clinical sort of stand-in media. This is at 1.25 mg per mil, which is above the ancestors. You can see the ancestors in most cases can't grow at all. The evolved ones can like grow a little bit better, but this is really like not much to write home about. I want to contrast that with the drug fluconazole. So again, this is our like sort of darling drug that we like study all the time. This is that same type of experiment. This is, I think, the first experimental evolution study done in Canada albicans. This is from Leah Cowan's PhD work with Jim Anderson, published in JBAC in, I think, 2000. Yeah, in 2000. So this is 6 replicates, same sort of trajectory, increase the drug concentration, see what happens, log MIC, and they all do what you want them to do, which is increase in MIC over the course of the experiment. Yeah, they evolved in fluconazole. We were like, well, do the same thing in boric acid. It did not work. And it turns out that things evolve or adapt to fluconazole even when you don't necessarily want them to. So this is sort of very new work from my lab, a grad student Aruni. I'm working in collaboration with Teresa O'Meara's lab at the University of Michigan, where we did mutation accumulation in Candida albicans, and Teresa had done it, had a postdoc do it in YPD in her lab, and then they left, and she was like, hey, you like evolution? You want to help me analyze this experiment? And I was like, sure, but let's do it again in fluconazole, and to see very much like what the Sharp lab looks at, like, let's look at the mutation spectrum under neutral populations in fluconazole against YPD, because there's this thought that fluconazole is potentially a mutagen. We know it causes aneuploidy. It can cause aneuploidy at higher rates because it sort of messes things up with cell cycle and budding. Let's look to see if we get like neutral aneuploidies higher and like actually like quantify that. And so Aruni, so in Teresa's lab, they'd done this in YPD, so just a rich medium, 50 transfers, and Aruni was like, I'm going to do it again in fluconazole. That same thing. We have this like cool method, I think it's cool method to look at competitive fitness. So she takes some ancestor, which has been marked to fluoresce GFP, and she mixes that with our evolved cells, and then we can just, kind of hard to see, but we can basically count fluorescing and non-fluorescing cells. And so I'm going to show you actual competitive fitness of how they're doing head to head with the ancestral and the evolved cells. And so she actually ended up repeating this mutation accumulation multiple times. And what she sees is that even in very low levels of fluconazole, when you look at competitive fitness in fluconazole, they've all gotten better. This is just 10 transfers in lower levels. This is sort of their MIC. This is competitive fitness measured in the drug level that they were involved in. So this is the YPD lines. This is sort of the neutral expectation of like they've not gotten better because we've bottlenecked them repeatedly and not necessarily given them scope to adapt. But it turns out even transferring single colonies in even low levels of fluconazole below your MIC, so a sub-inhibitory concentration, they're all getting better within the time frame of these experiments. So very, very different than boric acid. All right, so to go back, boric acid, maybe it works really well because there is little capacity to adapt to it. I didn't show you the experiment in glabratus where everything went extinct immediately, and no matter what we did, we could not get them to keep growing. I think it's really cool though, like we did get them to grow in higher levels, but then when we pulled them out, put them in the freezer, take them back out of the freezer, that seems to have gone away. So next is to like repeat those experiments and then do a bunch of things on the populations straight out of boric acid at the end of the evolution experiment. And so this old, small yet complex, sort of ubiquitous molecule seems to be a highly effective treatment. And I guess the like human side of this is that this shouldn't require word of mouth or Amazon reviews. And so as I talked to a few people today, one of the things I'm gonna start doing to like, I think you get tenure and you're like kind of allowed to do what you want to do. And so one of the things I'm gonna start doing is like trying to be a sociologist. And so looking at like health equity questions and like how are people getting to see the specialist? What does that path look like? What are people doing who don't live in the city? And I think there's just sort of lots of interesting and really important things we can learn from just talking to people who have these recurrent symptom cycles, and hopefully we'll be able to learn something that we can maybe feed back the biology and then feed back onto the human side of things. All right. So I just want to kind of end by saying I think Candida albicans is very cool. I think there is tons of open questions about what makes a species behave how it behaves in different niches and different populations in different contexts. And I think with this like era of sequencing capacity, we can kind of do what I call like gumboot ecology in microbiology in a way that we've never been able to do before. I thought these were going to be really easy questions to answer, but it turns out that they're not. And that there's very few like sort of comparable studies. So like just even looking at how much variation there is in a population, to me, should be something that's been studied many, many times, but it hasn't been. And so we're really excited to sort of take some of these new technologies that our lab and other labs are developing in this space to really get at what is the fungal population, how many species are there, how often are they there, what is the temporal stability of their diversity of their genes, and to sort of answer these really fundamental questions in evolutionary biology that then feed back into, I think, the clinical side of things as well. So with that, I will say thank you again for all the wonderful conversations I had with people today, to my lab, to the people who participated in our clinical work. None of this is possible without people volunteering to do things for us, clinical collaborators, sequencing, and funding. And thank you all for listening. Yeah, they hit it so well. Yeah. Yeah.
But when people have like chemical speculation and then sort of relatedly, how widespread effect? Is it only in big growth? Does it affect other fungi?
Yeah. Great question. Great question. So we haven't yet started the like microbiome work, but we are going to ask that question essentially. Same with fluconazole. There's other fungi in the microbiome. And so does it wipe everything out? We don't know essentially, but it's an antimicrobial. So like it almost certainly also affects the bacteria. And so I think I was talking about this with someone today, like we pull out albicans or cobratus or whatever, but it could be that other things are also, sorry, this is going to be annoying. It could be that other microbes are also influencing inflammation. So yeah, to be determined, I would say. And I think, yeah, yeah. And it seems to be this, and yeah, right. I mean, it does seem to it certainly like knocks down the yeast population. Also knocks down the bacterial population. So, and Matt's lab just published a really beautiful paper showing us how maybe we can do better microbiome work. And so we're excited to also play with the cell sorters that we have in my lab. Yeah. Well, no one can make money off it because it's just like in the like it's not, you can't patent it. You can't, like, it's not like Pfizer can suddenly slap a label on it and charge a bunch of money. There's also conflicting reports about whether or not it's harmful. So in Germany, you're not allowed to prescribe it for anything because there's some old study that shows it could be harmful. But like, no, right? It's one of those things where you have to ingest like kilograms of it to hurt you. It's a women's health problem. That's my answer. Yeah. Follow-up question on that. Yeah. What about I mean, like Vanessa is prescribing it. Yeah, so that's why we started studying it in the first place, because she was like, I prescribe this drug. But it's not like, if you can't go into the pharmacy and just buy it over the counter, but you can go on Amazon and buy it over the counter. So it's not like it's regulated. It's just, and fluconazole often works really well, right? And is really well tolerated by the body. So for most people, that's totally fine. And fluconazole is off label now, and you can buy it fairly cheaply in the pharmacy. Clotrimazole works for lots of people. Like, there are other alternatives for these one-off infections. It's just its recurrence. And that, I haven't talked to other clinicians about what they're doing. And that's part of what I want to do, is like talk to people outside of Vanessa's little circle of existence to be like, do you even know about this? Can you prescribe it? Like, what do people do in rural Manitoba who aren't being Vanessa? I have no idea. What do you do if you can't afford over-the-counter antifungal drugs? Do you just, like, what do you do? I don't know. Yeah. Yeah. So we looked, again, we thought that they would have adapted. And so we just put them straight in the freezer like we always do with the idea, well, then we'll pull them out and we'll do all the things. And so we didn't do anything at the end of the experiment. And so what Ola is going to do in the summer is redo that whole experiment and then like look right after. We pulled them out of the freezer into like low levels of boric acid and looked at morphology, and we don't see anything obvious. Like the only really things to look at are like yeast versus hyphae. Like that's the thing to look at, and we don't see anything. And they're all growing as yeast, but yeah, we don't see anything obvious. But we haven't done the right experiment yet. Yeah. Yeah, so I just submitted a grant to look at epigenetics. I mean, that is sort of my question. So maybe I didn't hit this enough. Is like, I'm super guilty of viewing things as like everything is adaptation, but like a lot of things could just be acclimation too. And so I think we need different methods maybe to study that. And that's something we're really interested in developing, but it's challenging, right? Like, how do you take a swab and not mess it up enough so that you're still getting the signature of being in the body on that cell population? And so we've been thinking about it enough to write grants about it, but yeah. Yeah. No, so Ola, because we got this kind of like null result, we didn't end up, she, did something else. So we haven't done any sequencing. Yeah. But yeah, we could. Yeah, that's a good question. So it really seems to be, well, so we see them increasing in the like we see the rectal incidence increasing for sure when we have high symptoms or high vaginal populations also seem to correlate some of the time with high rectal populations. We don't see high populations at other body sites. So when I'm just showing you like zeros and ones, check marks of X's, we almost have never seen more than like 3 isolates at any other body site. So like the oral and skin positive ones are like 1 to 3 usually. But we have to do the like actual like ASV or like molecular, because we know we have data that shows that there is some detection limit below which you won't get a colony and above which you will. And so just seeing an X doesn't necessarily mean they're not there. Yeah. Yeah. I don't know the data off the top of my head, but like it's been known as an antimicrobial since like over 130 years, something like that. I don't know like the first use, but like there's a paper from like 1981 that talks about whether or not we should be prescribing it. And so that's 43 years ago. So before then, yeah, long time. Yeah. 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 Right. What are the differences? Yeah. Yeah. So we're working with Joe Rhodes and Norman Van Rhyme to look at Africa genomics. And really this and like we don't have a good reference genome yet for it. So if you map it against albicans, we don't see much differences, but we want a good reference genome before we start asking exactly what's different. Yeah. Yeah. And there's a few that aren't vaginal. Like we see them in a few other contexts. They really do see vaginal. Yeah. Yeah. Say that again. Running the prescribed boric acid. Yes. Yeah, so we haven't done enough, like we just don't have a population, like we don't have the sample size yet to dig into. I don't think we have a population size yet. And it's also like, so we get we ask them to fill out these extensive questionnaires. They've all had like 10 plus cycles of symptoms in their lives, and their like drug histories are extensive. And so I don't know that we can like tease apart. We have avoided that just because I don't think we have the statistical power necessarily to say anything, but we would really like to look at what happens to the like microbiome right after prescribing boric acid versus fluconazole. I think we can ask that question for sure, and we're really excited to ask that question. Yeah.